So first off, homeostasis is the ability of our bodies to maintain a stable internal conditions so that our bodies can function properly. And it's very important that our bodies can stay at the right temperature, that we can keep the right pH, that we can maintain the right blood pressure. Because if we can't keep these things homeostatic, then our body's not going to work very well. And so homeostasis works through something called feedback loops. And first, we're going to talk about negative feedback loops. So here is a chart. And this is where your body wants to be. This is the set point right here, the set point. And let's say if we're talking about temperature, that's about 98 degrees. And that's where our body wants to be. And so as we feel cold outside, let's say we're cold, and this is a temp on the side, temp. And then down here, we're talking about time. If we start really cold, our body's gonna be like, oh, we gotta warm up. And so our body's gonna warm up, and we're gonna be too hot. And our body's like, oh, we gotta cool down. And so our brain's gonna send messages to our body to like, oh, it's time to cool down. And so that's called a negative feedback loop. And when the brain realizes and gets signals that it's like too hot or too cold or other stimulus for blood pressure or something, it'll like reverse it. It'll warm up or cool down depending on where we are compared to the set point. So we're always going to stay within this range so that our bodies can maintain a good level, good conditions. So there's something called positive feedback, which is different than negative feedback. And that's saying if we're up here, our body would keep going up. And if this is happening for temperature, that would not be good. That means our body's not working right. So usually positive feedback loops are not good for our bodies 95% of the time. But the 5% of the time, our body actually might be using positive feedback for something good. For example, birth uses positive feedback with labor and contractions. So the way these feedback loops work is you start out with a stimulus. Okay. And an example of a stimulus might say, let's say it's cold outside. It's cold. The next step is we have receptors on our body that can sense conditions outside or inside our body for different things. So if we feel that it's cold outside with the receptors on our skin, we're gonna start the negative feedback pathway. So the receptors are gonna work through an afferent pathway. And afferent means that it's going to the control center, to the thing that tells us where our body should be at, what the homeostasis levels are. So the receptor goes through the afferent pathway, and this usually includes neurons and blood. So it's like just nerves or the blood signals that we can tell the brain that something needs to change, or the control center, which is usually the brain. Control center. And then the control center is like, oh, this is what's wrong. This is what we need to fix. Through an efferent pathway, we're going to send a message to our effectors pathway, to our effectors. And this is just the brain telling our body what to do to correct different levels in our body. And so if we're talking about the cold specifically, we're going to have cold receptors on our skin that are going to go up neurons. That'll go to our brain in the hypothalamus and tell us, hey, we're a little cold. And so then it's going to send through neural pathways back to our muscles and other parts in our body. And if we're cold, our muscles are going to actually start shivering. And so that's like an effector, is shivering muscles. Or also the constriction of blood, especially in our exterior limbs, so that our blood can stay warmer. And we'll be able to maintain heat more in the core of our bodies. So that's what's going to happen when we're cold. So yeah, that's how a feedback pathway works. And let's see. So next we're going to talk about something else. We're going to talk about protein-mediated transport. So, well, first of all, the first transport we're going to talk about isn't even protein-mediated. We're just going to talk about simple diffusion. The way simple diffusion works is if you have some molecules in the extracellular fluid, ECF, and nothing in the intracellular fluid, that's a C, or at least less, it's 5-3. If this is the case, we're going to have a gradient, which means there's a difference in concentrations on these two different sides of the cell membrane. So by diffusion, the gradient will cause these molecules to cross through the membrane and into the cell. And for simple diffusion, these molecules have to be small, non-charged molecules for this to work. Because if they're too big, they're not going to just go through the membrane. And this is made out of the phospholipid bilayer. And so these lipids, they're okay with non-charged molecules going through them. But if these were charged, lipids would say, no, you can't come through. And so that's how simple diffusion works. Molecules like this could be like carbon dioxide or O2. They can just go in and out of cells as they please. And they'll always follow the concentration gradient. Next, we're going to talk about another type of diffusion called facilitated diffusion. Facilitated. Okay. So the way facilitated diffusion works is it's also going to be working with smaller molecules, but these ones could be charged as well. So that's a difference that we're going to have here. So just like simple diffusion, there's going to be a concentration gradient happening. So there's going to be more molecules on one side than the other. And this is what diffusion really is it's just diffusing across a membrane because of a gradient. So these will also cross through, but since they're positively charged, they will not be able to just cross through the membrane. So there's going to be, if they're negatively charged, that'll inhibit that as well, if they're just charged at all. So there's going to be channels on the cell that then allows the diffusion to occur. And there's different types of channels that have different purposes. And so first, we're just going to talk about pores. When you think of a pore, this is what a pore is. It just allows these molecules to pass through. But some pores are gated, and that means it's a carrier-mediated diffusion. So I'm going to erase this. So some pores are voltage-gated or voltage-gated channels, which is caused by charges on the inside and the outside of the cell, which we'll talk about later. So when the inside of the cell becomes too positive or too negative, this channel will open or close, then allowing these molecules to come in or out. So an example of this works with sodium. If this gets too positive on the inside, this channel will open. Like, just imagine if this is like a lid right here with a positive charge. this gets too positive inside, it's gonna like push against that lid, right, and open it. It's gonna allow these to come in. This isn't exactly how it works. It's a bit more complex with like a spring type alpha helix in here that'll open and close depending on the charge in here. And it's got calcium ions in there. Anyways, moving on. I'm not gonna get too in depth with this right now, but this is a facilitated diffusion for voltage gated channels. There's also ligand gated channels. With a ligand gated channel, these channels are going to actually have a spot for a certain thing to attach to, a ligand, and then that will allow the channel to open and close. So if we have the correct ligands, it'll open and allow diffusion to occur. So, for example, hormones could allow this to happen. And then the other type of gated channel is the mechanically gated channel. And this could exist on your skin. For example, if you touch something, like just the movement, the vibration of touching something will open these channels and allow molecules to enter your cell. And specifically molecules that will start an activation, what potential? What's it called? So it'll allow you to feel things through that. It's going. So we're gonna go back to simple diffusion real quick and mention it. Simple and facilitated. With simple diffusion, I'm gonna get a graph right here. Concentration and rate will be up here. Rate. The higher the concentration, the higher the rate. There's no limit. If you just keep adding more and more molecules to the outside of the cell, the faster and faster they'll go in. So the higher the difference in concentrations, that affects the rate. However, facilitated diffusion is a bit more complicated because you can increase concentration, and it will go faster and faster. There's going to be a maximum limit when it's fully saturated, that the rate can't go any faster. Because there's only so many pores that allows the molecules to go through them. So it's going as fast as it can, and once you get to a certain concentration, a certain difference. So wanted to note that for our diffusion. All right, so there's two more ways that our cells allow molecules to get into them. And the first two I talked about are powered by kinetic energy, which comes from the concentration gradients, just the desire of the molecules to spread out and to take up all the space that they can. And that's how diffusion works, and just fill the empty spaces. However, with the next two types I'm going to talk about, it's not fully dependent upon kinetic energy. It actually uses ATP as well. So we're going to talk about primary active okay, and this is a type of transport, primary active transport. So first off, what's going to happen is we're going to talk about a specific kind. So there's going to be ATP on the inside of the cell that will allow molecules to come in and out. It's going to power this transporter. And we're going to have 3 sodiums coming in. Sorry. And we're going to have 2 potassiums coming out for every ATP. And this is like this is a pump, basically. So it actually pushes both of these things against the gradient. Both the previous items we talked about go with the gradient, both types of diffusion. But this is not diffusion because it actually goes against the concentration gradients. And that's why it requires ATP to make this happen. So this powers it. And then secondary active transport. Well, before we get to secondary, this pump creates a positive charge on the outside of the cell, in the extracellular fluid, and it's going to give the inside of the cell a negative charge because you're pumping out. Sorry, I did this wrong. It's actually 2 potassium going in and 3 sodium coming out. My bad. So there's more sodium that's getting pumped out for every 2 potassium, and so you're going to create a positive charge on this outside and a negative charge on the inside. So that's what this pump does, and it creates this electric potential in our cells. So that's where the secondary active transport comes into hand, or comes to mind. Secondary active transport. You're going to already have this charge, and these are all going to be sodium ions out here that want to come back in because you just pump them all out there. And it's not even the charge that's driving this, it's just that there's a concentration gradient we're creating here because there's so much sodium out here, and there's potassium in the inside. And these want to come in so bad because we've just been pumping them out and they can't get back in. And so we can actually utilize their desire to come in by secondary active transport. So there's a special pump that when the sodium comes in, because it wants to come in, it's going to take something else in with it. It's like, hey, we can go in together. And the way this channel works is if it'll like conform to both the sodium and another molecule, and then only together can they get through. That way we can force larger molecules inside with the sodium that's powering its way inside, or powering its way through the membrane into the intracellular fluid. And this is called symport because this and the other molecule are both going in together. They're going in the same direction. But secondary active transport doesn't just do symport, it can also do antiport. The sodium still wants to go in, but it can also force other molecules out of the cell that we don't want. So this is called antiport because they're going in opposite directions. And that's a way for our cell to get rid of waste and other molecules that we don't want anymore through secondary active transport, specifically antiport. And so I think that's all that I have to say about diffusion and active transport. So we're also going to, real quick, I need to mention competition. That's kind of has to do with actually the facilitated diffusion, where there's channels that want to come in. And there's molecules out here. They want to come in through here. First, I have to say that there's specific channels for specific molecules. Like these stars out here, if this was like huge glucoses, they're not going to come in through the same pores that are reserved for sodium. they're going to have their own special pores and channels that allow them to come in. And so there's a specificity with all these pores, and it allows our cell to control exactly what's going in and what's coming out. It's not just oh, here's a gate, anything can go through whenever. For every pore that I've mentioned and every channel, including the voltage gated channels and the ligand gated channels and mechanically gated channels, they only allow a specific molecule to come in that'll have a specific effect on the cell. And so it's important that we note that. And there's also something called competition, which means that, like, for example, if you have a toxin, certain toxins can come in and block the hole so that we can't get certain things to diffuse. And I just wanted to mention that. Okay. Finally, we're going to talk about osmosis. Osmosis. So we've already talked about diffusion of molecules, but osmosis is the diffusion of water, because there's some molecules that will not pass through the membrane, the cell membrane. And so we're going to have the external cellular fluid out here and the internal cellular fluid over here. And first off, let's talk about sodium. So usually sodium doesn't have channels that just allow it to come in. Usually they can only come in through the pump. Well, actually the pumps pump it out. They can usually only go in through special voltage gated channels. At least when it comes to neurons and nerves. But we're going to have these sodium molecules out here. And we have a little bit of sodium on the inside too. Okay, so start off, osmosis, or also known as osmolarity, is related to molarity. And molarity is the concentration of molecules in a specific volume. So if we're going to add sodium chloride, NaCl, to a solution, might get like 150 moles. Let's see here. 150 millimoles. Okay, it's fine. 150 millimoles of NaCl in a solution. And that's what molarity is. However, osmolarity takes more than that into account. Osmolarity takes into account like all of the molecules and particles, all the particles in a solution. So sodium chloride actually will split into two when it enters water. And so we're actually going to get more like 300 milliosmoles. Can't read that. Milliosmoles for that. So that's what osmolarity is, and it's very, very important for the function of cells. And so in the human body, we're just going to say that the osmolarity is 300 for sodium in the extracellular fluid and in the intracellular fluid. Yeah. So if this is 300 and this is 300, they're the same concentration, even though we know that there's going to be more sodium on the outside. They're going to have the same concentration because there's much less fluid on the inside of a cell. So if we inject someone with say a saline solution that's also 300, it's going to stay the same, 300 milliosmoles, because the concentration won't change. You're adding water and more sodium, so it's going to be the same osmolarity. And if we're adding 300 specifically, that's going to be an isoosmotic solution because it's the same concentration as the inside of the cell. And nothing will happen because it's the same concentrations. However, if we were to add a hypoosmotic solution, let's say 150 milliosmoles, we're just going to inject this into someone, into their extracellular fluid. This would lower the concentration of the ECF. Let's say it lowers to 200 for whatever reason. At this point, there's going to be a higher concentration of sodium ions on the inside of the cell. And water in the extracellular fluid is going to be like, oh man, you want to be closer to these sodium ions. And water, H2O, is going to actually go through the membrane, and this cell is going to expand. It's going to swell. And that's what's going to happen when you add a isoosmotic solution to the extracellular fluid. If there's a hyperosmotic solution that we're adding, let's say we added 400, and this total went up to, 350. Water is going to go the other way. Water inside the cell is going to want to go to where there's more sodium because this is a higher osmolarity out here compared to in here. And so the sodium will leave the cell. Not the sodium, I take that back. The water will leave the cell and cause the cell to shrink, or crenate is the fancy word for shrinking cells due to osmolarity. And that's what's going to happen if we have a hyperosmotic solution that we inject into the extracellular fluid. So that's what osmolarity is, and that's what it has to do with osmosis, the movement of water across the cell membrane. But we also have to think about tonicity. Tonicity is a little bit different than osmolarity, but they're very related. With sodium, I believe the tonicity is actually the same as the osmolarity. If the osmolarity is isoosmotic, the tonicity will be isotonic as well. However, if we're working with something like dextrose, this will be very different. So if we were to add an isoosmotic solution of the dextrose, it would still be 300 milliosmoles, right? Because that's what osmolarity is. However, dextrose, which is basically glucose, will actually go through the cell membrane and become digested. It'll be destroyed and go through the cellular processes that exist inside the cell. And so we're actually going to wind up with 0 milliosmoles outside the cell, and this concentration is going to be super low. And that's not going to be good because at that point, the water is going to leave the cell. Hold up. Water is going to enter the cell because there's a higher osmolarity on the inside. And it's going to keep going because there's nothing out here, and the cells will continue swelling and probably explode. And so that's what's going to happen with dextrose, 5% dextrose, which means it has a 300 milliosmolarity. So that's what tonicity is. So even though we had an isoosmotic injection of dextrose, it would be hypotonic, because really we're going to end up with less that's going through. And you can also end up with hypertonic solutions and isotonic solutions. So yeah. Yeah, that's all I have to say. Thank you.